Good morning, every online audience. The next question speaker, I am pleased to welcome Professor Makoto Higuchi to our joint symposium on nuclear neurology and cardiology. Professor Higuchi holds an office of department head, served in Department of Functional Brain Image, National Institute for the Quantum Science and Technology. He has excellent experience to make research on brain imaging and published many papers in past year. His present speech title is Novel PET Image for the Neurodegenerative Disease. Please join me in welcoming Professor Higuchi.
Hello, everyone. I'm Makoto Higuchi from National Institute for Quantum Science and Technology, or QST, in Chiba, Japan. So my talk today is focusing on the development of the novel PET imaging technologies to visualize the core pathologies of the neurodegenerative diseases. So here's my COI disclosure. So let me quickly overview tauopathies and the alpha-synucleinopathies, initially from the viewpoint of the neurodegenerative dementias, which are composed of the Alzheimer's disease, or AD, frontotemporal lobar degeneration, or FTLD, and the dementia with the Lewy bodies, or DLB. So these illnesses are clinically diagnosed primarily based on the characteristic symptoms. But there are actually remarkable overlaps of the symptomatic manifestations among these disorders, as exemplified by the memory impairment in all of these dementing illnesses and the Parkinsonisms in progressive supranuclear palsy, PSP, corticobasal degeneration, CBD, and the DLB. So PSP and the CBD are neuropathologically defined as tauopathies, and the DLB and the Parkinson's disease in the alpha-synuclein pathology continuum. So synuclein depositions are also pathological features of multiple system atrophy, or MSA. So the presence or absence and the localization of tau and the synuclein deposits would provide the objective indices for the diagnosis and the differentiation of diverse dementing and the Parkinsonian syndromes. So tau imaging agents are now available for both non-clinical and clinical research and the trials. So the first generation tau PET probes, including C11 PBB3, F18 THK 5351, and the F18 T807, or AV1451, have been performing nicely in capturing 80 type tau aggregates in the brains of living cases. However, there have been difficulties in imaging FTLD type tau pathologies with the use of these radio tracers. So for example, PBB3 could be rapidly metabolized after intravenous injection, resulting in quite low brain entry and a small dynamic range for the target visualization. And the THK compound have shown high cross-reactivities with the monoamine oxidase B, which is highly expressed in reactive astrocytes. And the T807 exhibit only modest binding to FTLD type tau fibrils. So for imaging AD and the non-AD type tau pathologies by PET, we need to consider the fact that the tau is composed of 6 isoforms synthesized by alternative splicing of the transcripts of a single gene. So the isoforms are classified into 3 repeat and 4 repeat tau species according to the number of the repeat domains. The AD is characterized by the deposition of the all 6 tau isoforms. In FTLD syndromes, so Pick's disease in Pick bodies in Pick's disease are neuronal tau inclusions composed of 3 repeat isoforms only. So the aggregation of the 4 repeat tau isoforms in neurons and glia is a feature of the other FTLD disorders such as CBD and the PSP. So the diversity of the tau isoform composition may result in the differences in the reactivity of these fibers with each of the tau imaging agents. In our recent work published in Neuron, we developed a new probe, PMPBB3, for capturing a wide variety of tau aggregate species in AD and FTLD. Along with the transgenic mouse models of tau pathologies with high contrast. So indeed, in the mouse model developing tau fibers, tau PET imaging with the PMPBB3 intensely illuminated the tau deposits in the neocortex and the hippocampus, in contrast to the minimal binding of PBB3 to these aggregates in the same animal. So a head-to-head comparison of PMPBB3 and the PBB3 was also conducted in 80 patients. So by looking at the scale bars here and here, so you may acknowledge that the PMPBB3 could yield a much higher contrast for tau deposits than PBB3 without the noticeable off-target binding in the brain parenchyma. So a notable feature of PMPBB3 is its capability of illuminating the disease-specific tau topologies in each of the scanned individuals. So tau depositions in the neocortex and the hippocampal formation of AD, so subthalamic nucleus, thalamus, and the basal ganglia of PSP and the CBD, and gray and white matter of the motor cortex of CBD. Could be sensitively captured by this radioligand. It is also noteworthy that the PMPBB3 PET could capture 3-bit tau aggregates in the frontal cortex of a Pick's disease case for the first time. So the presence of the Pick bodies in the brains of this particular subject was thereafter confirmed by autopsy assays, which will be shown in my talk later. So our collaborators at the Changgung Memorial Hospital in Taiwan demonstrated the similarities of the topologies of brain atrophy and the PMPBB3 positive tau depositions in AD cases. So indicating that this radiotracer captures toxic tau species leading to the neuronal loss. So the spreading of tau pathologies following the so-called Bragg staging from the limbic to the neocortical regions was trackable in AD. And the intensification and the expansion of the radio signals from the subcortical structures to the neocortex were traceable as a function of the PSP severity. So these findings may be indicative of the propagation of the tau pathologies through specific neuronal circuit, but more compelling evidence will need to be provided by a longitudinal assay of the same cases. So AD cases could be completely separated from controls without any overlaps, yielding the sensitivity and the specificity of 100% in the use of the limbic and the neocortical SUVR values, which are measures of the radiotracer retention. And the close correlations between the neocortical SUVR and the disease severity assessed by CDR sum of boxes was observed in the AD group. Similarly, PSP cases could be separated from controls with little overlap, yielding the sensitivity and the specificity approximating 95% in the use of the SUVR measures in the subthalamic nucleus. So moreover, a close correlations between the SUVR in the subthalamic nucleus and the severity of the disease scored by PSP rating scale was found in the PSP group. So therefore, PET imaging with the PMPBB3 may offer an objective index for the propagation of the progression of the AD and the PSP. So then biopsy and the autopsy assays of the brain tissues derived from the CBD, PSP, and the Pick's disease patients who had undergone the PMPBB3 PET scans provided the clear evidence for the presence of characteristic 4-repeat tau aggregates in PSP and the CBD cases, and the peak bodies consisting of the 3-repeat tau fibrils in Pick's disease case. So these data support the in vivo reactivity of PMPBB3 with the diverse FTLD type tau assemblies. So our research group at the Huashan Hospital in China has also made publications on the accurate discrimination between PSP cases and the healthy controls using PMPBB3 PET data. So they have also found a good correlation of the subcortical radioligand retentions with the PSP rating scale scores. So indicating the utility of this imaging technology for staging PSP in an objective manner. So for applying a PMPBB3 PET to the diagnosis of the diseases in the real world clinical practice, we have developed an automated machine learning based analysis of PMPBB3 PET data for the diagnosis of AD and PSP. So briefly, elastic net machine learning can be performed with the use of the probe retention measures in multiple regions of the interest. And the AI can be trained to maximize the performance of the so-called AD tau score or PSP tau score in the diagnostic discriminations. So the PSP tau score calculated in each subject is capable of separating PSP cases from healthy controls and AD cases with reasonably high accuracy. And the AD tau score is also capable of separating AD cases from healthy controls and the PSP cases with almost perfect accuracy. So here is the list of the tau PET imaging agents available for clinical studies and practices. So as compared to the first generation probes, the second generation probes are capable of detecting AD type tau deposits with the high contrast. But apart from PMPBB3, these new traces originating from the AV1451 and may show no or modest binding to the non-AD type tau aggregates. So among these second generation tau PET probes, PI2620 developed by Piramal is also supposed to detect the 4-repeat tau aggregates in PSP. So a recent report by a Keio University group has documented that an enhanced retention of the PI2620 is confined to the subcortical areas, such as the midbrain, in contrast to the widespread radio signals yielded by PMPBB3, so in the same individual. These two PET probes may react with the distinct binding components in PSP type tau aggregates. And the PMPBB3 could be more suitable for capturing widespread tau accumulations. So our partner, Taiwanese venture company, Aprinoia, is currently conducting the phase 2 clinical trials for PMPBB3, also known as APN1607 or fluozolotau in the US, Japan, and Taiwan. So this year, they have also launched a phase 3 clinical trial for this probe in China. So hopefully, fluozolotau will be available for clinical sites as an approved diagnostic agent in 1 or 2 years. Following tau imaging probes, so the development of the alpha-synuclein imaging agents are now at the clinical stage. an earlier study led by Tohoku University group showed that the alpha-synuclein aggregates in oligodendrocytes, known as glial cytoplasmic inclusions or GCI, could be detected in the brains of MSA cases by a PET tracer termed BF227, which was originally developed as an amyloid beta ligand. However, alpha-synuclein pathologies in the vast majority of the MSA and the DLB patients were undetectable by BF227. So then we also demonstrated by autoradiography and the fluorescence staining of the brain slices, so that our tau PET probe, PBB3, displayed weak cross-reactivity with the GCIs in MSA. So again, this ligand was incapable of detecting alpha-synuclein deposits in most of the cases with the DLB and the MSA. So in fact, the reactivity of PBB3 with the alpha-synuclein assemblies, so in human brain tissue, quantified as an IC50, was more than 50 nanomolar, indicating that the ligand binding was more than 30 times weaker than the value required for a practical imaging tracer. Then we have recently reported the generation of the bimodal optical and the PET probe for alpha-synuclein pathologies, and the preprint of the manuscript on this work is placed on the bioRxiv site. As most of the beta sheet ligands are self-fluorescent, so their reactivities with the protein aggregates could be evaluated simply by fluorescence staining of the brain sections with test compounds. So in this study, we generated a new class of the chemicals by substituting a CC double bond in the linker structure of PBB3 with the triple bond. And this relatively small modification was sufficient for 40-fold increase of the affinity for the human alpha-synuclein aggregates, and 60-fold increase of the specificity for DLB alpha-synuclein versus AD tau deposits. So the linker substitution alters the twist angle of the backbone structures, which could be probably critical for the binding of these compounds, so with the different beta sheet structures in tau and the alpha-synuclein fibrils. So we then inoculated the recombinant alpha-synuclein fibrils into the striatum of a wild-type mouse to be used as a model of disseminating synuclein pathologies for in vivo characterization of the new imaging agents. Again, the new probe C0505 is self-fluorescent and is usable as an optical tracer. So here we just labeled the blood vessels with the red fluorescent dye SR101, and then in line with the postmortem finding on the spreading of the alpha-synuclein deposits from the striatum to the neocortex. So intravital two-photon microscopy after intravenous injection of the C0505 demonstrated the disseminating synuclein aggregations, reaching the cortex at 4 weeks. So followed by the transient decline at the 6 weeks. So under re-intensifications at the 8 weeks. Longitudinal assessments of the individual synuclein aggregates also revealed the intraneural propagation of the fiber formations retrogradely from the axon to the soma, and the loss of neurons burdened with the alpha-synuclein aggregates. So we then radio-labeled C0505 with the F18 and the PET imaging of mice at 4 months after the cyanocolline fiber inoculations into the bilateral striatum demonstrated increased radiotracer retentions in the striatum and the neocortex relative to controls, suggesting that the alpha-synuclein depositions macroscopically accessible by PET. Similar to mice, alpha-synuclein fibers were inoculated into the unilateral caudate and the putamen of marmosets, and the dissemination of the synuclein pathologies from the injection site to the neocortex and the midbrain and the contralateral counterparts could be tracked by longitudinal PET assays with the F18 labeled C0505. So in parallel to the synuclein imaging, dopamine transporter PET scans of these marmosets showed the impaired dopaminergic synapses in the ipsilateral striatum and the midbrain, indicating the neurotoxicity provoked by the disseminating synuclein pathologies. So in vitro autoradiographic and the fluorescence labeling of the brain sections with the C0505 demonstrated the ability of this probe to clearly detect the alpha-synuclein pathologies in the MSA and the DLB brains with moderate to heavy synuclein burdens. So therefore, it is anticipated that a large subset of alpha-synucleinopathy cases would be diagnostically accessible by PET with the C0505. We are currently working on the development of the C0505 derivatives in collaboration with the several Japanese pharmaceutical companies in a public-private partnership alliance. So a new PET probe dubbed SPAL-T06 is being tested in a clinical study and has exhibited sufficient brain uptake followed by the rapid clearance, yielding the low background signals at the late phases of the PET scan. So taking advantage of this pharmacokinetic property, we have succeeded in high contrast PET imaging of alpha-synuclein aggregates in living patients with different MSA subtypes. So the details of our latest findings with this probe will be presented in the next chance. So to summarize, so there have been a small molecule beta sheet ligands capable of capturing the tau aggregates of AD, PSP, and CBD types in living brain, allowing the diagnosis, differentiation, and the staging of these dementing and atypical Parkinsonian disorders on a neuropathological basis. And then new class of the chemicals derived from the tau probes has shown a high affinity for alpha-synuclein fibers. So in vivo PET studies of murine and non-human primates models demonstrate the utility of these imaging agents for pursuing the disseminating alpha-synuclein depositions and possibly their suppressions by candidate therapeutics. So the alpha-synuclein imaging agents are applicable to the intravitreal microscopic detection of the individual synuclein aggregates in mouse models and would be utilized for the identification of the mechanisms linking alpha-synuclein deposition and the neurodegeneration, so leading to the neuronal loss. And the first in-human PET study of PD, DLB, and MSA cases with the new alpha-synuclein PET probe, which is named F18 SPAL-T06, has been initiated in August 2021. And then we are now actually being successful in high contrast, detection of the MSA type alpha-synuclein aggregates in those living cases. So finally, I would like to thank all the co-workers and the collaborators listed here. So I also appreciate your kind attention. Thanks a lot.
I'm grateful to the Professor Higuchi for your excellent speech and sharing new robust research for us. The current time for online audience to ask a question to Professor Higuchi. If any question online audience. There are 4 questions online. Question 1, Professor Higuchi. This is my question. Many thanks, Professor Higuchi. Based on your experience, the PBB3 has high contrast for AD tau lesions and more robust ability to capture wide range tau pathologies. Can the second generation tau PET probe completely replace the first generation tau PET probe?
Thanks a lot for raising such a great question. So, can you hear me?
Yes,
Okay.
it's online, it's here.
Yeah. So I guess in the field of the AD research and the clinics, probably the second generation tracers, including our tracer and the Merck's tracer, may just replace the position occupied by the first generation tracer like AV1451. But at the moment, AV1451 is the sole tracer, so getting the approval by the regulatory agency in the US. So still, it may just, have the overwhelming roles in the clinical situations. And there are lots of accumulating data about, for example, longitudinal analysis of the tau deposits assessed by PET with the use of the AV1451. Those data are still lacking with the use of the second generation tracers. I think more accumulating evidence would be necessary for the substitution of the first generation tracer with the second generation ones. And in the field of the PSP and some other non-Alzheimer's disease tauopathy field, I guess our tracer PMPBB3 has already shown some kind of the primary roles in the assessment of the such patients. So for the diagnosis, differentiation, probably the staging in a cross-sectional and the longitudinal manner. Now we are in collaboration with the pharmaceutical companies for the use of such a tracer for pursuit of the the tau retention along the course of the disease-modifying therapy. So I think more evidence will probably need to be collected still, for PMPBB3, but that's the situation now.
Oh, thank you. Thank you, Professor Higuchi. Second question is from the Lincoln Lu professor. And we see some tau PET positive in amyloid negative dementia subjects. Do you have any comment on the diagnosis or outcome for these subjects?
Yeah, thanks, Kun Lu, for such an important question. So I guess, of course, this is probably kind of the topic. So especially about the so-called primary age-associated tauopathy, which is tau deposition without the amyloid deposition in association with the atrophy of some brain areas, including the hippocampal formation. So some people with the PART, primary age-related tauopathy subjects, shows some cognitive declines, but that only can slowly progress. So I don't know if such people should be just treated with some disease-modifying treatment at such an early stage. So probably, I guess, longitudinal topic studies of those so-called pod subject would be necessary to just follow the time course of the tau pathology along with the clinical progressions. If there's some group of the people with pod showing the relatively rapid clinical progression, so then we need to consider the intervention, early therapeutic interventions in such a subject based on the probably tau PET imaging findings.
Another question, Professor Higuchi, according to your lecture about the new human tracer of F18 STLT06, which has utility of different MSA subtype for alpha-synuclein aggregates. Is it also available for detection of Parkinson's disease and Lewy body dementia? Thank you.
Okay, so yeah, thanks for the great question. Actually, we have already done the clinical PET studies with the use of the so-called our new novel alpha-synuclein PET probe, and we have succeeded in the high contrast detection of the alpha-synuclein aggregates in MSAC and MSAP subjects so far, and now we are applying this probe to non-MSA type alpha-synuclein pathologies, including DLB and PD. I guess only a subset of the DLB subjects seems to be positive for our tracers. So because of the lack of the abundant alpha-synuclein aggregation or a relatively low affinity of our tracer for DLB versus MSA type alpha-synuclein aggregates, we do not know the reason of the, for example, failure of the, detecting all the subjects. So only a small subset of the DLB subjects seems to be positive. And the more challenging thing is capturing alpha-synuclein aggregates in idiopathic PD subjects, because alpha-synuclein aggregates seems to be confined to really tiny anatomical structures like substantia nigra of the midbrain. And now we are trying to capture such an challenging material. So we will probably provide our experiences in the use of this new tracer in diverse alpha-synuclein pathologies quite soon in a publication or in a conference. So that could be soon.
Thank you, Professor Higuchi, and we're happy to see the future results soon. And another question from Dr. Wang Qichang. The results from the C05 related studies are very impressive, especially in the murine model. The alpha-synucleus can be seen a couple of weeks after the treatment. Since he's also performing some experiments using the alpha-synucleus preformed fibrils, is it how many amount of the PFF is you applied in the study? He would like to know, and in which brain areas? Thank you.
Okay, so the question is about the actually non-clinical, model of the alpha-synuclein dissemination. So I do not remember the exact amount of the preformed fibrils inoculated into the striatum of the mice and the marmosets. So, but now that can be just, those, the experimental protocol are already shown in our preprint, uploaded in bioarchive sites. So I think you may just search for our names or C0505 in the bioarchive sites to just, find out our manuscript. So documenting such a non-clinical experiment using animal models. So if you just have some difficulty in finding that, just let me know. So I may just address such a question or related issues.
Thanks, Professor Higuchi. The third is by Professor Lincoln Lu. I hope we will have the opportunity to work with you for the alpha-synuclein PET study in Taiwan. It's a good.
Yeah, thanks for the comments. So it will be also my pleasure to collaborate with your group. So just like our collaboration, so regarding the use of the PMPBB3, so that will be really fruitful, and we could probably hopefully get good data. So in capturing the alpha-synuclein aggregates in diverse neurodegenerative disorders. So that's my hope.
If there are no other questions, many thanks, Professor Higuchi. I appreciate you taking the time for the expert speech and the discussion. Next time, I hope to have the opportunity to invite you in the live conference. Thank you. Thank you very much.
Yeah, thanks a lot. My pleasure to join the conference.
Thank you. Thank you.